Hello, welcome back to our final review video in preparation for your exam, and maybe you're doing this for homework as you're going through that chart, but this review video is going to be all about cell organelles. Now, as we are learning organelles, I'm going to tell you a little bit about me when I was learning organelles the first time. I kind of just thought of them as separate entities that all did their own thing, but in reality, they are all working together to make a functioning cell. So you have to remember that they're all communicating to each other, and we're going to see how that works throughout this PowerPoint. And when we did that activity in class, that's organelle speed dating activity. Yeah, it was a lot of fun, but you don't really get to see how all the pieces are working together to make a really functional cell. But of course, that is going to start at the nucleus, which is going to be this circular, round structure found near the center, and it's going to contain and protect the DNA, which will be in condensed form in chromatin. Now, inside the nucleus is this dark region called the nucleolus, and the nucleolus is going to create ribosomes. We're going to see why that's really important later on. We're going to talk about ribosomes soon, but yes, you need to know that. You need to know both of these, not just the nucleus, but the nucleolus as well. Okay, now, this is all covered by something called the nuclear envelope. This is going to be a double membrane found around the nucleus. And you can imagine, well, we're really protecting that DNA super well. The problem is that's making it less accessible, so we need access to that DNA. Remember that, yes, the DNA codes ultimately for proteins. It codes for mRNA, and then the mRNA codes for proteins. But that mRNA is going to have to get to a ribosome, which is not going to be permanently in the nucleus. So it's going to have to travel through something called nuclear pores, these holes in the nuclear envelope. And these regulate what comes in and out of the cell. Now, we talked about this when we talked about endosymbiotic theory. It's not directly related, but we talked about it on the same day. And remember that because we have this compartmentalized area for our DNA, we have an extra area that we can regulate between transcription and translation. And this gives eukaryotes a huge advantage to prokaryotes because we are able to, yes, regulate, and that means that it's not all or none. If I get a signal, I can regulate based on that signal, whereas something like bacteria is going to basically use the signal and then generate the protein right away. So that extra regulation is a huge advantage to eukaryotic cells. So I told you that the ribosomes are important, and they are. Remember, they are a hallmark of the cell, one of the four, and so they must be pretty dang important, and they are. I studied ribosomes for a summer. I studied the mitochondrial ribosome once in a summer during my college, so I'm a big fan. They are going to be two subunits, one large and one small. Now, interestingly, rRNA, or ribosomal RNA, acts as a catalyst. This is weird because when we think of catalysts, you should be thinking enzymes, and you're right. Enzymes are catalysts. In fact, the vast majority of catalysts in the human body and really all life are going to be enzymes, which are proteins. Don't forget that. But this is one exception where this rRNA, a nucleic acid, is acting like a catalyst. This is very weird. Sometimes we call it a ribozyme instead of an enzyme, but it is a nucleic acid that is acting like an enzyme, which is very bizarre. Now, this rRNA catalyzes the reaction to form peptide bonds between amino acids. So it catalyzes the reaction to form a protein. And so that means that the ribosome is going to be important for protein synthesis. Okay, so the ribosome is clearly important for protein synthesis, and I told you that the ribosome is in every cell, which tells you that proteins are pretty dang important. You cannot have a cell without proteins. You can't have really life without proteins. And so if it's so important, we should probably understand how it's getting from one place to the next in the cell. We should understand what they do, and a lot of what we study in this course is kind of studying just proteins. So when we start talking about this thing called the endomembrane system, in a lot of ways it is talking about how do we deliver proteins from one area of the cell to another. And so we have specialized organelles that are going to be involved in that. Okay, so the organelles that are involved are going to be the endoplasmic reticulum, both the smooth and the rough, the Golgi body, several types of vesicles, lysosomes and peroxisomes, and they are going to connect basically from the nucleus. You can see over here in the yellow this time, the nucleus in yellow is basically figuring out how can we get stuff from the nucleus ultimately over to the membrane. Maybe it's getting spit out, and it's the other way too. So we would call it the secretory system if it is spitting it out, but the endomembrane system is very, very, very important. So let's talk about it. So the endoplasmic reticulum, it surrounds the nucleus, and it is going to be a bunch of membrane channels that are connected directly to the nuclear envelope. So this is one way that we can get sort of stuff out of the nucleus and into the cell, which is very, very important. And we have two types. We have rough ER, which is going to be connected to the nuclear envelope. And you can see on these purple, all these purple dots, these are representing ribosomes. So ribosomes are directly connected to the rough ER. So sometimes you'll hear people say that the rough ER makes proteins, and that's not totally accurate. The thing that's making the protein is the ribosome, but the ribosome is connected to the rough ER, making it look rough. Okay, the smooth ER is different. It doesn't have any ribosomes, so it shouldn't be making any proteins. It is still connected to the nuclear envelope and the rough ER, but it's famous for producing lipids. So the way I remember this, and it's a little silly, but it actually has still worked since when I was taking AP Bio, I always think, okay, things that make my skin soft are often oils. Oils are lipids, and so when I say smooth ER makes my skin smooth, it's a little silly, but it does help me remember. It can also break molecules down, and it can send vesicles to the Golgi body. So we're going to talk about vesicles in just a second, but this is important. So remember, we are getting things from the nucleus, from ribosomes, proteins, and we're trying to deliver it to different parts of the cell, usually the membrane. Okay, so that term vesicle is an important one. Vesicles are going to be these membrane-bound organelles, and they contain materials inside them. So you can have a vesicle that is holding something, typically a protein inside, and it is going to get delivered somewhere eventually. But it's not going to know where to get delivered right away. So the smooth ER, the rough ER, is going to send this vesicle to the Golgi. The Golgi is the next one we're going to talk about, but I first want you to understand how a vesicle forms. So a vesicle is going to form from physically pinching off from a membrane. So these things are basically transient. They are going to exist for a while, and then they're going to deliver to somebody else, to some other organelle. So they pinch off, potentially, literally pulling off membrane from the ER, containing whatever it needs to contain, and then it's going to deliver that vesicle to the Golgi first, and then after that, the vesicle will travel elsewhere. But the idea here is that they are delivery vehicles. Now, they're not permanent, they go away, but they are delivery vehicles that are containing something in the middle, and they're going to bring it to certain organelles. So like I said, the first stop it's going to make is typically the Golgi body. So the Golgi is going to receive transport vesicles from the ER. And then it's going to modify whatever is in that vesicle. So like I said, it's usually proteins. But the question is, when we make a protein at the ribosome, how does it know where to go? Because proteins are made for the mitochondria at the ribosome, they're made for the lysosomes, they're made for the peroxisomes, which we're going to talk about later. But how does it know where to go? Well, it needs to stop at the Golgi apparatus first. And the Golgi apparatus is going to modify that protein, and it's going to tell the vesicle where to go. So oftentimes these modifications to these vesicles are going to be glycoproteins, which are going to act like a tag and say, okay, this is a mitochondrial protein package. This is a lysosome package. And the Golgi is going to be the thing doing that. Now, you don't need to worry about terms like cis-phase and trans-phase. You don't really need to understand how it moves through, because even that mechanism isn't totally understood by scientists. And At one point, if you are really interested, you'll learn more about it in upper level college biology courses, but that is not appropriate for today. But I do want you to understand it's going to receive a vesicle that is physically pinched off from something like the ER. It is going to modify that vesicle because that vesicle fuses into the Golgi. It's going to be modified, and then that's going to tell the cell where the contents of that vesicle need to go. Okay. And so these extracellular proteins from the ER, they're going to be released from the cell when vesicles fuse with the membrane. So we might have something that is actually designed to leave the cell. So it's not important to this direct cell, but it needs to leave it. And so it'll pinch off from the ER and then travel to the membrane. But remember, it makes that stop at the Golgi first. It's going to stop at the Golgi. The Golgi is one of my favorite organelles, by the way. Okay. Now we also have membrane-bound organelles. So what we just talked about was those endomembrane organelles. Now, those are all membrane-bound, but these are also membrane-bound, but they're less important for delivering things around the cell. So the first one here is lysosomes. So lysosomes are going to be they're membrane bound, and they are produced by the Golgi apparatus, and they contain digestive enzymes. They're very, very, very important for digesting cellular contents. They are going to destroy non-functioning organelles. They can eat invading pathogens. So if there is a pathogen that enters the cell, it can get destroyed by the lysosome, and it can destroy other things. Now, when I say destroy, that doesn't always mean it's a bad thing. It's designed. So we have something called apoptosis. I'm going to try to write it. My handwriting is not very good when I'm using my mouse. I apologize. Apoptosis, which is programmed cell death. And the lysosome is very important for this process. You should know that. Star it. Know that it's very important for apoptosis. Okay, and so I want you to think about how would this relate to life? Because remember, AP, always about application. Well, actually, your lysosomes are very, very important for your embryonic stage in pregnancy. So your hands, when you're in the womb, are originally webbed. And so they actually have webbing between the fingers. And so the lysosomes are things that are the organelles that actually are digesting that webbing between the fingers. And when it fails, we actually have webbed fingers. So there are, a handful of people, not a huge percentage. Obviously, we don't see a lot of people with webbed hands, but it can result in webbing. And so we can see that here, with these two fingers that sort of got combined because the lysosomes clearly failed in this area. It is not a horrible thing. This is going to still be a relatively normal life, but it is certainly something that is not planned. Okay. We also have something called peroxisomes. So peroxisomes are going to be, once again, membrane-bound organelles, and they still also contain enzymes. They are mainly helpful for breaking down fatty acids and dangerous chemicals. That's the piece that I want you to really understand, these dangerous chemicals. Why? Well, because a lot of our reactions, especially in metabolic reactions, they are going to produce free radicals. And free radicals are pretty unhealthy because, as you can imagine, this word radical tells you that it's a highly reactive, I guess it is a chemical. So it's a highly reactive chemical that can sort of just take in electrons, they can just steal it, and they're super reactive, and so they win almost every time. And that's pretty dangerous. We don't want a lot of radicals in our cells because they would just sort of mess everything up. And so these oxidases are able to convert these radicals into something called peroxide, hydrogen peroxide. And then a second step here, a different enzyme, is going to this enzyme catalase is going to convert hydrogen peroxide into water, something we can easily handle. So hydrogen peroxide isn't a great thing to have in your cell, but once we get it to water, that's doable. So 2 steps here that happen. First enzyme is an oxidase, the second one is a catalase, and it is going to ultimately reduce the amount of free radical in our cells. So thank you, peroxisomes, because we would be dead without you. Okay, so how does this relate to life? Well, this actually directly relates to a condition called Zellweger syndrome. So Zellweger syndrome is a rare inherited disorder that is really quite sad. It has a very high mortality rate. It results in severe cranial abnormalities, visual and hearing impairments, and seizures. And it's caused by a defect in the PEX gene, the PEX gene. And that defect results in a reduction of functional peroxisomes. Well, we just talked about the function of peroxisomes, which is basically clearing out free radicals. And so if we have reduced the number of peroxisomes, we now have an accumulation of free radicals, which can result in pretty severe conditions and, once again, that high mortality rate. So these are a couple of people who suffer from Zellweger syndrome. Unfortunately, it is very, very, very deadly. Okay, so I'll be very honest with you. Vacuoles are one of my least favorite organelles. They're just a little boring to me because they're basically just storage sacs. They are not vesicles. Vesicles, remember, are typically our delivery vehicles. Vacuoles are much more permanent members of the cell, and they store things like food and water. For plants, this is very, very important. They are just a big storage thing, and they actually can give a little bit of structure to the plant cell as well. Okay, one of my favorites, the chloroplast. Why do I love the chloroplast? Well, because without it, we could not exist. And I know you're saying, well, they're not even in humans, and you're right. But without them, life couldn't exist. So, chloroplasts are membrane-bound organelles. Maybe you remember, they are actually double membrane-bound. And remember, they're double membrane-bound because when they were taken in by an early eukaryotic-like, sort of prokaryotic-like cell billions of years ago, remember, through phagocytosis, it actually got its outer membrane from the eukaryotic-like cell that ingested it, and it had its original cell membrane because it was originally a free-living cell. So remember, it's double membraned. It is filled with pigments. We call it chlorophyll. And it is, of course, very important for a very, very special process called photosynthesis, where it uses solar energy to synthesize carbohydrates. We will talk a little bit more about the structure of chloroplasts when we get to that unit, Unit 3. So you don't need to worry too much about things like the thylakoid and the stroma. But do know that it is a double membrane bound. It actually has an electron transport chain, which allows it to make ATP. That's important too. Write that down. Wink, wink, nudge, nudge. It has an electron transport chain, which we're going to talk about a little bit more in the next unit, but it's also a similarity to something we're going to talk about in a second. So chloroplasts, they are double membraned, and they have their own electron transport system, much like the mitochondria. So the mitochondria is also double membraned, much like the chloroplast, but it's a little bit smaller. Now, remember that it's double membraned because, just like with the chloroplast, endosymbiosis, a large cell once ingested something that was basically a mitochondria and gave it its second membrane. So the mitochondria was once free living, so it had its own cell membrane, then it was ingested and it got a second one. So it is double membraned, and it is very, very important that we understand that it does also have an electron transport chain, which is going to be important for producing ATP. So the chloroplast has that as well. So yes, the chloroplast does produce ATP, just not as much as something like the cell, the mitochondria. So it produces ATP because it has a very efficient electron transport chain, which we are going to talk a whole lot about when we get to Unit 3. But you do need to know that now. You need to know that that is a major similarity between the mitochondria and the chloroplasts. You want to look for other similarities as well. For example, DNA, presence of DNA, mitochondrial DNA, chloroplast DNA. There are ribosomes. All that stuff is true of both chloroplasts and mitochondria. The major difference, of course, is that plants have both, whereas humans and animals only have mitochondria. Okay, the other piece to this puzzle about mitochondria is looking at this electron image of a mitochondria. We can see that it is highly folded. So this is super, super, super folded. These are all little folds. You can see them. Now, you might be asking why. Well, it is increasing the amount of space to do the third step of cellular respiration, which is the major producing step of ATP. That step is the electron transport chain. So once again, we'll talk about it a lot in Unit 3, but I do need you to know that now, that's why we fold our mitochondria so tightly, because it gives more room for the electron transport chain, which means that we get way more ATP out of something that is highly folded as compared to something that would just maybe be one round in a circle around the edge of the organelle. Okay, so there is a structure called the cell wall that we don't have, meaning humans and animals don't have, but actually it seems like most organisms do. 90% of bacteria cells have cell walls. Theirs is made of peptidoglycan. Plants have cell walls. Theirs is made of cellulose. And then fungi also have cell walls, and it's made of chitin. And there might be more. I'm just not totally aware of them. But cell walls are very present in a lot of organisms, but not in us. We don't have a cell wall. We have a cell membrane, and so do plants, so do bacteria, but we do not have a cell wall. So these are going to be made out of carbohydrates and proteins. Famously, carbohydrates, peptidoglycan is I guess it's a protein and carbohydrate. Cellulose for plants is a carbohydrate. And it's going to be very important for maintaining shape and protection for the cell. So remember when we started talking about water potential and osmosis, the cell wall was the thing that was giving pressure against the cell. So it ended up creating a pressure potential. So that kind of made for more fun math problems, right? And then we have the cytoskeleton. The cytoskeleton is going to be a structure of basically fibers around the cell, and it gives our cells some strength. So it's very important for our cells. It's important for plant cells. Now, the one thing is there is something similar to it in bacterial cells, but it is not quite the same in the sense that it's the identical proteins. So we have something called actin and microtubules, which we'll see in a slide, but it's adjacent to that in prokaryotes. So it's very important for the shape, and it's also important for movement. So we see in this final bullet point that it has a dynamic structure, and it'll rearrange as needed. So it's a little hard to see in this image, but you might be able to see all these little dots. These little dots are basically tiny little monomers of tubulin, basically. So we have what we call tubulin subunits that will form a large microtubule. But these can get rearranged, and we can shorten it and we can extend it based on the cell need. Same with the actin filament. So we're able to shorten it and extend it as the cell needs, depending on what is trying to happen in the cell. So it's very dynamic, it's not rigid, and it's very important for structure. Okay. So microfilaments, that's this thing called actin. I'm not going to worry too much about this. I do want you to know that actin and microtubules are important for the cytoskeleton, but you do not need to know the structure of these things. Just know that they exist. You can study that a lot more when you take upper level biology. And the same thing with intermediate filaments. So these intermediate filaments, they're much like rope-like structures. They are also, once again, part of the cytoskeleton, and that's important. But you do not need to know the structure. Same with microtubules. They are going to be made up of tubulin, which becomes very important when we get into Unit 4. But again, you do not need to know the structure here. If you're very interested, I strongly recommend studying biology, and you'll get to take some of these fun classes later on. Okay, so that brings us to the end of the review. I know that was a lot of material, and maybe you didn't get that much material in class, so please make sure that you are knowing this material, it's making sense to you. If you need to, rewatch this a couple of times. I've also made a Quizlet for you, so access that Quizlet, start learning these organelles, learning their functions, learn where they're important, and what processes they're very important, and try to think big picture, try to think about why and how these organelles work together, rather than thinking that everything is just isolated in one spot, but in reality there's a lot of communication within the cell. Okay, good luck. I wish you the best of luck on this next exam. Ciao.